Assalamualaikum warahmatullahi taala wabarakatuh and good evening to each and everyone. I'm Bansilba Mardea and tonight I'll be discussing the continuation of behaviorism of proteins, enzymes, mechanism, and control.
So let's proceed to determining the essential amino acids in the active sites. The serine residue at position 195 is required for activity of chymosubstine. In this respect, chymotrypsin is typical of a class of enzymes known as serine proteases. So, this determines identifying which amino acids in an enzyme's active site are essential helps understand its catalytic mechanism. For instance, in chymotrypsins, a serine residue at position 195 is critical for catalysis, classifying it as serine proteases. Determine essential residue. Researchers use chemical labeling techniques. Chymotrypsin is inactivated when it reacts with disopropyl phosphofluoridate, which forms a covalent bond with the active site serine residue. This method shows that only specific amino acids participate directly in catalysis. Such studies are vital for designing enzymes, inhibitors, and understanding biochemical pathways. Biochemical connections allied health. So, proteases, a diverse family of enzymes, catalyze the hydrolysis of proteins through a shared mechanism involving an active site, which means proteases are enzymes that catalyze the hydrolysis of peptide bonds in proteins. They are divided into classes based on the active site reduces involved, such as serine proteases, example, yung chymotrypsin, and cysteine proteases. Papain or papain derived from papaya is a cysteine proteases commonly used as a meat deterrizer and in wound healing. It uses a cysteine histidine catalytic aid where the cysteine residue acts as a nucleophile to attack peptide bonds, and histidine aids in proton transfer. This mechanism underscores the importance of specific amino acid residues in the enzyme's catalytic power and biomedical relevance. So next is chemical reaction involved in enzyme mechanism. Enzyme mechanism often involves specific chemical process such as acid-base reaction, covalent bond formation, oxidation reduction reaction, and metal ion catalysis. Enzymatic mechanism rely on fundamental chemical process that facilitate substrate conversion in products. So we have common types include the acid-base catalysis. This is where amino acid side chains donate or accept protons to stabilize reaction intermediates. So covalent catalysis, where a temporary covalent bond forms between the enzymes and substrate, lowering the activation energy. So oxidation reduction or redox reaction, where electrons are transferred between molecules, often mediated by cofactors. So metal ion catalysis, where metal ions stabilize charges or participate directly in redox process. So, this mechanism ensure that enzymatic reaction proceed rapidly, selectively, and under psychological conditions, distinguishing them from non-biological catalysis. Mechanism of catalysis. This refers to how enzymes speed up chemical reactions. The mechanism of catalysis describe how enzymes accelerate reactions. Enzymes do this by lowering the activation energy required for the reaction. They stabilize the transition state, properly align substrates in the correct orientation, and create microenvironments favorable for the reaction. Some enzymes use acid-base catalysis to transfer protons, while others use covalent catalysis to form transient bonds with substrates. So next is the active site and transition states. The active site of an enzyme is a specialized region where substrates, the molecules that undergo chemical change, bind and react. The structure of the active site is precisely shaped to fit the substrate, much like a lock and key. Although the modern endosphete model explains that the enzyme can slightly change shape to accommodate the substrates better. Within the active site, specific amino acid residues interact with the substrate through hydrogen bonding, hydrophobic interaction, or ionic bonds, helping to stabilize it and orient it correctly for reaction. This transition state represents the highest energy, most unstable intermediate during the conversion of substrate to product. Enzymes work by stabilizing this transition state, effectively lowering the activation energy, the energy barrier that must be overcome for a reaction to proceed. By doing so, enzymes significantly increase the reaction rate without being consumed in the process. Establishment of this transition state is key to enzyme efficiency and specificity, ensuring biological reaction occur rapidly and under mild psychological conditions. Enzymes or catalytic antibodies are laboratory-engineered antibodies that act like enzymes. Unlike natural enzymes, enzymes are produced by designing antibodies that bind to a transition state analog, a molecule that resembles the high-energy transition state of substrate during a reaction. Once the antibody binds to this analog, it can catalyze the same reaction as an enzyme by stabilizing the transition state of the actual substrate. Enzymes represent a fascinating intersection of immunology and enzymology, demonstrating how immune molecules can be engineered for catalytic activity. The whole potential in medicine and biotechnology, including applications such as targeted drug detoxification, therapeutic enzyme replacement, and biosensor development. For instance, enzymes can be tailored to degrade harmful substance or catalyze reaction that natural enzymes cannot perform efficiently. Biochemical connection allied health catalytic antibodies against cocaine. Catalytic antibodies or enzyme can be engineered to neutralize harmful drugs such as cocaine. These antibodies catalyze the hydrolysis of cocaine into inactive products before the drug reaches the brain. By preventing cocaine from interacting with neural receptors, such enzymes offer potential as therapeutic agents for cocaine addiction or overdose. This approach represents a novel biochemical strategy using the immune system specificity and enzyme-like catalytic power to target and degrade toxic substance directly in the bloodstream. Coenzymes are small, non-protein organic molecules that assist enzymes during catalysis. They act as temporary carriers of specific atoms, electrons, or functional groups that are transferred in enzymatic reactions. So we have the common coenzymes include, these are the NAD and FAD and coenzymes. NAD is transfer electrons in redox reactions. NAD is this is the nicotinamide adenine nucleotide. So FAD naman is flavin adenine dinucleotide. This is another electron carrier in oxidation reduction processes. So coenzyme or CoA carriers acyl groups in metabolic reactions such as the citric acid cycle. Most coenzymes are de- derived from vitamins, for example, vitamin B, and are essential for metabolism and energy production. Without coenzymes, many enzymes would be inactive or making them crucial for maintaining circular function and functional end life. Next is biochemical connection, environmental toxicology, catalyst for green chemistry. So, in green chemistry, enzymes and coenzymes are used as biocatalysis to promote environmental friendly chemical processes. Unlike traditional industrial catalysis, enzymes work under mild conditions and produce fewer toxic byproducts. This makes them ideal for sustainable manufacturing in pharmaceuticals, agricultural, and biofuel production. By replacing harsh chemicals with enzyme based systems, green chemistry aims to reduce pollution and energy use, demonstrating how biochemical principle can protect both health and the environment. Alzheimer's disease is a chronic neurodegenerative disorder that leads to memory loss, cognitive decline, and behavioral changes. Biochemically, it is associated with the accumulation of abnormal proteins in the brain, notably beta amyloid plaques and tau tangles. Tau is a protein that stabilizes microtubules in neurons. In Alzheimer's, tau becomes abnormally phosphorylated, causing it to form tangled structure that disrupts cell communication. So, apolipoprotein or APOE plays a role in lipid transport. That ApoE4 variant increases the risk of Alzheimer by affecting amyloid beta clearance and brain repair mechanism. So, researcher into enzyme activity, protein folding, and biochemical regulation help scientists understand and seek treatments for this devastating condition. So, I guess dito na mag-end ang aking presentation. Again, I'm Bansilva Mordea and thanks for listening. Bye!